I'll talk about some basic FMRI and analyzing of FMRI with AFNI data and what I'm working with there currently. So FMRI is like functional MRI is a non-invasive technique for studying brain activity, and during the course of an FMRI experiment we acquire lots of brain image, and it's like the subject is performing a set of tasks and we just change the measured signal between individual images to inference like which part of the brain image is working, which part of the brain is active and how they are reacting. Each image consists of like 100,000 of voxels, and it's like we just in our brain volume we just divide it into 100,000 of voxels, and each voxel corresponds to a spatial location and has a number of associated with it that represent its intensity. For example, we can choose like one voxel is like number is 39, and we just find out like how the intensity of this region is behaving with the change of like performing different tasks and how it's reacting to the task. And during the course of an experiment, several hundred images are acquired. It's like one every 2 seconds, so it's like one in every 2 seconds we get like the whole total image behavior, like full brain activity we can find out in 2 seconds. Then after that we again perform the same thing and image is acquired accordingly, and we find out the behavior of our brain. And we can see like suppose one voxel of our brain image we can find out, and we can find out that how the intensity of that region is changing. So it's clear like with the task like suppose a clapping of hand or like finger tapping, and with that performing task the intensity of suppose one location we can see the change of that location, how it is changing with the task. So the most common approach towards FMRI users is the blood oxygenation level dependent. So cold FMRI measures the ratio of oxygenated to deoxygenated hemoglobin in the blood, and it is like it does not measures the FMRI directly, neuronal activity directly. Instead it measures the metabolic demands of active neurons. That is like when some region of our brain is active, then it needs the oxygenated blood. So oxygenated blood flows to that region, and with that we can find out like how the activity is changed in the brain region. It's like hemoglobin exists in two different states, like one which is oxygen with the hemoglobin, and that is oxyhemoglobin, and this is diamagnetic, and the deoxyhemoglobin is paramagnetic. And for cold FMRI, it takes the advantage of the difference in T2 star between oxygenated and deoxygenated hemoglobin. So when there is like the concentration of oxyhemoglobin blood is increasing, that means the FMRI signal will be strong. And when it decreases, when the deoxyhemoglobin deoxyhemoglobin is increasing, that means the signal will be going from the strong to weak. And the changes in the MR signal triggered by neuronal activity is known as the hemodynamic response function, HRF we can call it in short. And as the neuronal activity increases, so does metabolic demands for oxygen and nutrients. That means like the neuron, it needs the oxygenated blood, so it just goes in peak. And overcompensated in blood flow reduces the concentration of deoxyhemoglobin and tips the balance toward oxyhemoglobin, and this leads to a peak in board signal about 4 to 6 seconds. After reaching the peak, the board signal decreases to an amplitude below baseline level. That means like it's like due to a combination of reduced blood flow and increase the blood volume. And we can see like this is the HRF or hemodynamic response function, and we can say like when suppose some task is done, like the hand finger clapping or something is done, then the signal goes to peak and then it again goes down. That means like when the oxyhemoglobin increases, the signal rises to a peak, and after that, after 5 or 6 seconds, it's like, and to go to that peak, it needs some time, like 5 to 6 seconds. This is called the delays. Like when the task is performed, it does not go to peak at that level, because for oxygen to flow that region, it needs some time to go to that region. So whenever we are clapping, like, so there is some delay, like we don't get it like at that exact time, so get it after some times. And fMRI data analysis is like lots of data. So each brain volume consists of like 100,000 voxel measurement, and each experiment consists of like lots of brain volumes. We check it like every 2 seconds, like how the brain is, how it is reacting and how it is acting with that. And the statistical analysis is like there is massive data problem, like lots of data, and the signal is relatively weak, and the data exhibits a complicated temporal and spatial noise structure. So we have like lots of noise in our fMRI. So the source of noise is like thermal motion of free electrons in the system. It can be like from the patient movement during the experiment, and like physiological effects such as the subject's heartbeat and respiration, or it could be like from low frequency signal drift. Low frequency signal drift is like if a person is not doing anything, it's like no work is performed, still like there will be some slow change of signal. So like nothing doing, but still there will be, so this could add the noise also. And finally, like what we are doing from the like analysis of this data, we want to find out like which region is active, so determine like which region of the brains are active during a specific task, and like if some regions are active, then we can find out like how they are reacting with, how they are connecting with each other. Like these are our tasks. And for analysis of this functional analysis of this data, we can use the AFNI. AFNI is like analysis of functional neuroimage. It was developed to provide an environment to fMRI data analysis, and it's a platform for development of new software. And it refers to both programs of that name and the entire package of external program and plugins. It's like important thing about the AFNI is like it allows the user to stay close to the data and view it in many different ways. We can find the data like how it is working in the like in different location, in different it's like in like different view, and it's one thing is like it allows other programmers to add features and that can interact with the rest of the package. And what I am doing currently is like I was working with AFNI. I was given some analysis of some data, and analysis of some AFNI data. Now I am just writing a code in C, so that like it is with like the real-time plugin of like in a real time is like whenever we are doing the experiment, we are getting the data just at that moment. So there is some noise. There is some noise and other things. So I was just given a task like to write a code in C and just to remove that. Like there is some predetermined like noise. We can find out like how is that behavior of that. And so like real time when we are getting the data, we can just directly get the smooth data, not like the raw data, so that we can find out like the behavior of the data and other things.